We will be continuing with Chapter 12, looking at transport across cell membranes. We're picking up here with a focus on active transport. In active transport, we will see that solutes are transported against their electrochemical gradient, and this requires an outside energy source, an additional energy source. I will focus on specific examples of two of these types of fuel. The first one we will look at is an ATP-driven pump, one that changes shape or undergoes conformational changes via phosphorylation, and the energy from this phosphorylation is what drives the transport of a solute from low concentration to high concentration. We will also look at a gradient-driven pump. In this case, this orange solute is still moving from low to high concentration, but at the same time, another molecule will be moving down its concentration gradient. A gradient-driven pump uses stored energy in a concentration gradient of one solute to fuel the transport of another solute against its gradient. A third energy source for active transport is light, although we will not be looking at a specific example here. In this slide, we see a very important sodium-potassium ATPase. It's a pump that creates the concentration gradients of sodium and potassium. You can see that we're going to move sodium out of the cell and potassium into the cell. This sets up these gradients where sodium is at a high concentration in the extracellular fluid and potassium is at a high concentration in the cytoplasm. So potassium being pumped from low to high, sodium being pumped from low to high, both against their electrochemical gradients. And this is fueled by phosphorylation. This slide details the conformational changes that are regulated by phosphorylation and allow for active transport of sodium and potassium. To understand the picture, it's helpful to focus in on the shapes. The authors of the text have simplified things by making sodium a circle and potassium a square. If we start off here in the upper left-hand corner, we can see a circular solute binding site for sodium. And sodium can come from the intracellular space and bind to the sodium binding site. This does not trigger a shape change. However, once the pump is phosphorylated, right, so we're hydrolyzing ATP, in this conformational change, the binding site changes shape so that it can bind potassium, and we also create an opening for sodium to be released. After sodium is released, potassium can now bind to the solute binding site. Binding of potassium triggers a release of the phosphate group, and that release of the phosphate is the switch to put the pump back into its original shape or conformation. Potassium is released, and the protein can begin a new round of transport. This process is also illustrated in the following movie.
Animal cells store energy in the form of ion gradients across the cell membrane. In the cytosol, the sodium ion concentration is kept low relative to the extracellular fluid, and conversely, the potassium ion concentration in the cytosol is kept high. Like water behind a dam, these gradients harbor potential energy that the cell taps to fuel cellular work. Animal cells use a membrane pump, called the sodium pump, to maintain these ion gradients. To begin the pumping cycle, sodium ions enter binding sites on the cytosolic side of the pump. Although there are 3 sodium binding sites on this pump, for simplicity only one is illustrated here. Pumping sodium against its concentration gradient requires energy, which is provided by cleaving ATP. ATP transfers a phosphate group to the pump in a high-energy linkage. Phosphorylation causes a dramatic change in the pump's conformation, so that the sodium ions become exposed and released outside of the cell. This action also exposes binding sites for potassium ions in the pump. Although there are 2 potassium binding sites, for simplicity only one is shown here. Binding of the potassium ions triggers release of the phosphate group and the return of the pump to its initial conformation. The potassium is then released inside the cell, and the cycle repeats. A complete cycle takes about 10 milliseconds.
So some transporters transport only one molecule at a time. This would be like the glucose transporter we covered in the last section, right? It moved glucose and moved it solely down its concentration gradient. Other transporters can move two solutes at the same time. It can move both of them in the same direction, which would be a symport, or it can move them in opposite directions. At the same time, the energy from moving one molecule down its gradient can be used to transport the other against its gradient.
Cells possess a variety of membrane proteins to ferry solutes across the membrane. One type of transporter, called a uniport, carries only one type of solute, selectively bringing it from one side of the membrane to the other. In contrast to uniports, coupled transporters carry two types of solutes. If both solutes are moving in the same direction across the membrane, the transporter is called a symport. In this example, the solute represented by the circle is carried down its concentration gradient, from high concentration to low. The energy released by the movement of this solute drives the movement of the other solute, represented by the square, against its concentration gradient, from low to high concentration. When the coupled transporter moves solutes in opposite directions across the membrane, it is called an antiport. In this example, the solute represented by the circle is transported down its concentration gradient, fueling the transport of the other solute, represented by the triangle, against its concentration gradient, that is, from low to high concentration.
Here we have the glucose-sodium symport. This symport is going to transport sodium down its electrochemical gradient from high to low, and glucose against its gradient from low to high. So sodium is moving with its gradient and glucose against, but they're both being transported in the same direction with respect to the cell, in this case, into the cell. So here we can look at the conformational changes that occur in the protein to allow this to happen. The sodium glucose symport can only close if it contains both sodium and glucose or neither. So we can see a closed conformation with no access to the extracellular or intracellular surface when there is no bound solutes or when both are bound. We are very unlikely to have both bind sodium and glucose in the interior of the cell. So sodium concentration is high outside the cell, and because you have that high sodium concentration, this is the only side where we are likely to have binding of both sodium and glucose. Sodium and glucose both binding triggers conformational change, and we get inward transport of sodium and glucose, sodium down its concentration gradient, glucose up its concentration gradient. In this case, glucose will always move in the direction of the sodium gradient. If I were to switch the electrochemical gradient of sodium and just pump a whole bunch of sodium into the cell, glucose would then move in the opposite direction. So the sodium gradient is determining the direction of glucose transport. So here's a question I pose. What would happen if the sodium concentration was equal across the membrane? Well, if that was true, then you would have equal transport, so no net movement of glucose. It would move inward and outward at the same amount. So pretty much whatever the gradient of sodium is going to happen, the same type of movement would happen with glucose. So we've gone over a glucose uniport in the previous lecture, a sodium glucose symport and then a sodium-potassium pump. All three of these transporters work together inside of your gut. So this is a picture of your small intestine, and we're picking out like this area right here to focus in on particular cells. And these cells have an apical surface that faces the inside or lumen of your small intestine. This is where digested nutrients and other things would be found, things like glucose. The blue here is the apical side of the cell, and then in gray is what's often called the basal lateral surface. So this would be facing extracellular matrix and then a blood vessel. Nutrients are going to travel from the apical side of the cell across the phospholipid bilayer into the cell, and then across the basal and lateral surfaces, and then into a capillary for collecting those nutrients. In this image, we are looking more closely at an epithelial cell and thinking about the concentration gradients and the transporters and the pump that are used to guide directional transport of glucose. So we can begin by looking at the glucose concentration gradient, which is depicted by arrows and the intensity of blue color on the right side of the image. You can see that we have high glucose concentration inside the cell and low glucose concentration outside the cell, both in the gut lumen and in the extracellular fluid. The sodium-potassium pump, circled here in red, creates a sodium gradient as well. What is the resulting concentration of sodium within the cell? We see that this gradient is opposite of the glucose gradient, where sodium would have a tendency to enter the cell when it moves from high to low. So we have a concentration gradient of glucose where it would tend to move outside of the cell by diffusion, and a concentration gradient of sodium where it would move inside the cell via its concentration gradient. Now we can add in the transporters to show how sodium is used to cause directional transport of glucose. At point A, at the apical surface, there is a sodium-driven glucose symport. And it is here that the sodium gradient, right, high to low, drives active transport of glucose against its gradient, right? We're going low to high, and glucose enters the cell. Without the action of the sodium-potassium pump, directional transport of glucose against its gradient into the cell would not be possible. Or if we had equilibrium in the sodium concentration, if it was equal inside and outside the cell, we would also have equal movement of glucose inside and outside the cell. So it is necessary to maintain a low concentration of sodium within the cell to allow for directional transport of glucose at the apical surface. So ultimately, the energy for import of glucose at the apical surface is due to the hydrolysis of ATP at the sodium-potassium pump. After glucose has entered the cell, it can then move across the basal membrane through the uniport. This is facilitated passive diffusion, where glucose moves from high to low concentration. The uniport, B, should only function at the basolateral surface. Diffusion barriers keep it from diffusing up to the apical surface. If the uniport were to be functioning at the apical surface, glucose would then diffuse out of the cell, going from high to low, because the uniport only works by facilitated diffusion.